Alright guys, I hope you're having a good evening, a good morning, or whatever time you're watching this. We are moving now from looking at how cells divide and how they multiply, and looking at what controls that. Why are we, taking genetic information, taking chromosomes, splitting them apart? What's really going on deeper inside of that chromosome? And so we want to look at something called deoxyribonucleic acid, and you may or may not have heard of that, but simply put, deoxyribonucleic acid is DNA, right? Oh, DNA. Sorry. Wow. DNA. Okay. And so I do expect you to understand and to be able to say and be able to spell deoxyribonucleic acid. We don't want to be scared of that word because it is a part of who we are. It's our genetic makeup. And so let's begin. Okay, we want to first review the role of DNA, and I'm sure that you can remember this. We know that DNA will help store, copy, and transmit genetic information into a cell. Store, copy, and transmit genetic information in a cell. So when we look at storing, its function and its importance is because of the differentiation that we want to have within each cell. If there's no differentiation or no genetic diversity within organisms like ourselves or other animals, there's a less chance of survival. And when we get to genetics and we get to natural selection, that'll make a little bit more sense. But we need differences between us so that our species can actually survive better, and so that we can have patterns of development. We have to have that information stored so that we can see the differentiation and patterns of development. For example, in the development of humans, bacteria, replacement of cells. For example, even a more specific example would be the dead skin cells, is that we have to have that pattern of development in order to slough off our dead skin cells and then replenish them. Secondly, looking at copying, it's important because each cell has to have the same genetic information. We may have genetic diversity, but we want to have the same, codes for hair color, the same code for, how tall you are, what sex you are, those sorts of things. An example of that is that we see that occurring during interphase of mitosis and meiosis. That is during the S phase of interphase, and that's when DNA is copied. And lastly, we want to have a transmission of DNA, and we'll talk about that within a couple of weeks, but it's a way that we can transfer that genetic information. We do see this within mitosis and meiosis, but we also see things getting copied with proteins so that we can create other things inside of our cells. It's not just transferring genetic information cell to cell, but it's also transferring and transmitting that genetic information within a cell so that proteins can come along and help produce other things that the cell needs. Now, let's look at deoxyribonucleic acid, DNA. We kind of talked about its importance, but it does help us assemble proteins, and it can help us to regulate various functions within our cells. And just so we know, back to our biomolecules, the nucleotide is the basic building block of DNA. And so let's take a look at that structure of DNA. It's the tiny building block. It is made of three things, and you can see that pictured here. You have a phosphate group. You have deoxyribose, which is where we get the D from in DNA. And it's just a sugar, right? Ribose, OSE. Remember, OSE refers to our sugars as a suffix. And then you have the nitrogen or the nitrogenous base. And this entire thing right here is our nucleotide. So you may want to write that down. Nucleotide. Okay? Now, the nitrogenous base, the nitrogen base, there are different types. There's 4 different nitrogenous bases, therefore we're going to have 4 different types of nucleotides because just changing this base is going to give us a different type of nucleotide. So we have the 4, adenosine, cytosine, guanine, and thymine, and you do need to be aware of those, and I think some of you may be. We do abbreviate them A, C, G, and T, but you need to understand that there is adenine, cytosine, guanine, and thymine. Now, we always have cytosine pairing with guanine. So in this example, we'll take a look at why this looks upside down. It is upside down for a reason, on purpose. But C always pairs with G, right here. And you always have adenine pairing with thymine, A pairing with T. They will never intermix. C will never pair with A. T will never pair with C. It's always A and T and G and C. So try to remember that. Try to help yourself remember those 4 names of the nitrogenous bases. And right here, something very special, and you can see that in the purple box, is that we have a hydrogen bond. Remember, hydrogen bonds are a somewhat weak bond, but they're a very favorable bond, especially when we need to break bonds apart later. And so it is really important for there to be a hydrogen bond, and I'm kind of talking about the why right now, but that hydrogen bond is important because later on we need to actually unzip DNA. We want to be able to break DNA here so that we can copy one strand, the one side of DNA, and copy the other strand of the other side of DNA, and zip it back together. So hydrogen bond is really handy because it's not it's not super strong, but it still does the job. Okay, so just to review, the sugar and the phosphate are going to be on the sides if you consider like a ladder. So if I were to draw some sort of ladder, okay, here's the sides of a ladder, or you can call it the backbone of the molecule. These sides here are going to be a phosphate and the sugar, a phosphate and the sugar, phosphate and the sugar, and then over here is going to be sugar phosphate, sugar phosphate. And the ordering of this has to do with the shape of DNA, which I'll talk about in a little while. But then in between, you're going to have your bases paired up. So phosphate and sugar, and then over here, you're going to have maybe A, right? Over here, you will end up having T. And these two will pair up. But if you notice, it's kind of angled. And so what's going to happen is we're going to get a special twist that we see on DNA because of that angle. It doesn't want to stay straight. And so what happens is, let's take a look here. You can see how it is staggered. In this picture, the way they drew it, it is straight across. But right here, just remind yourself, draw yourself a picture of a nucleotide. It's a phosphate, a sugar, deoxyribose, and our nitrogen. In this case, it's a C. C always pairs with G, but in the other way, it goes backwards. And I will talk about that in just a moment. So going back to why why it's shaped differently and why it was kind of angled. DNA has a double helix, and so this is a model of what it looks like, and it has to be twisted due to the way that things pair. And if you look back here, if this thing was tilted a little bit, you would understand why we get that twist, and we'll revisit that in the future. So that twisting, the way that things bond, the attractions that happen, between here, these are not going to want to be attracted to each other, so it's going to cause that to shift a little bit. These are not going to want to be attracted to each other, so they'll shift a little bit. And as you keep shifting things, that's how you kind of get this structure. You get that double helix. And the reason why one strand of the phosphates and the sugars was going in one direction and the other one was going opposite is that these strands are anti-parallel. They cannot go in the same direction, and so they run basically opposite of each other. And what that does is it allows for the nitrogenous bases to actually come into contact within the center of the molecule. It helps them to match and pair together a little bit better, and it has to do with the attraction of all the different chemicals that are surrounding it. And so what happens is we can start to label these ends, and labeling these ends is really important when we want to look and study DNA. And so we have a 5' to a 3', and so the 5' to a 3' always matches the 3', 5 to a 5', and I'll show you what that looks like in just a moment. So 5 to 3 is always in one direction, and then 3 to 5 always goes in the other direction. So if you were to just draw it, I'll just draw it real quick. If I drew it kind of this way, okay, you would have the 5 end here and the 3 prime end there. You put that little tick mark to represent prime, and then the 3 prime end runs here and the 5 prime end runs here. And then imagine this thing kind of twisted together, right? And then if I actually took it and twisted it, then that would be a little bit more of a 3D structure. Now the 5 prime end is always going to be on the phosphate end, so you always mark your little 5 there. So now let me go back and show you briefly here. I would be marking this as the 5 prime end. Down here, this is the 5 prime end. Up here, this is 3 prime. This is 3 prime. Okay? And then lastly, I just want to review a little bit with why there is this certain structure of the nucleotides and how come there is, how come they line up in such a manner. Well, there is a method to why. It is really important, the order of how it is aligned. And so what occurs is that the order of the nucleotides actually causes a genetic code. It's very unique. And so the more similarities that we have within the way the nucleotides are aligned, it shows us how much more related we are to certain animals or how distantly related we are to certain animals. So just to review, kind of just bring everything together, DNA codes for proteins. That's, part of what it does. And the order of nucleotides is going to be different for different proteins. So ultimately, the order of the nucleotides is going to code for what protein is going to be made. It's kind of like taking an alphabet and putting it together and scrambling it and making certain sentences, making certain phrases. Or maybe if I took the letters of the alphabet and I created a song and I had a verse and I had a chorus and I had a bridge. And it's kind of a weird analogy, but it is very specific, right? The way that the chorus is, you typically sing the chorus the same over and over and over. The verse 1 is always the same, verse 2 is always the same. And so those verses are kind of like the proteins, and the letters I'm putting in and the sentences I'm making are very specific. And so that's the order of the nucleotides that are creating that. So I know that's kind of a weird analogy, but maybe that could help some of you guys. So I think that's it.